শুভ সন্ধ্যা সকলোলৈকে আজি 15 মিনিট শিক্ষাৰ এই অনলাইন প্লেটফৰ্মত আমাৰ মাজত ডক্টৰ বাণীকান্ত শৰ্মা আছে ডক্টৰ বাণীকান্ত শৰ্মাৰ জন্ম হৈছিল ৰঙিয়াৰ কামৰূপত অসমত প্ৰাৰম্ভিক শিক্ষা ৰঙিয়াতে সমাপ্ত কৰি তেওঁ কটন কলেজত বেচেলৰ অফ সায়েন্স কৰিছিল কেমিষ্ট্ৰিত তাৰ পাছত মাষ্টাৰ অফ সায়েন্স কেমিষ্ট্ৰিত আইআইটি গুৱাহাটীৰ পৰা সম্পূৰ্ণ কৰি তেওঁ পিএইচডি কৰিছিল আইআইচি বেংগলোৰ ইণ্ডিয়ান ইনষ্টিটিউট অফ সায়েন্স বেংগলোৰ প্ৰফেসৰ জি মুগেশৰ তত্ত্বাৱধানত পিএইচডি সম্পূৰ্ণ কৰি তেওঁ পোষ্ট ডক্টৰেল ষ্টাডিৰ কাৰণে গৈছিলে ইউটি সাউথ ৱেষ্টাৰ্ণ মেডিকেল চেণ্টাৰ টেক্সাছত আছে ইউনিভাৰ্চিটি অফ টেক্সাছ ইন ইউএছএ আৰু স্ক্ৰিপছ ৰিচাৰ্চ ইনষ্টিটিউট ফ্লৰিডা ইউএছএ এই দুটা অনুষ্ঠানত তেওঁ সম্পূৰ্ণ পোষ্ট ডক্টৰেল শিক্ষা সমাপ্ত কৰি আৰম্ভণিতে শিৱনাথৰ ইউনিভাৰ্চিটিত এছিষ্টেণ্ট প্ৰফেসৰ হিচাপে কিছুদিন কটাই বৰ্তমানে তেওঁ নিউ কেমিষ্ট্ৰি ইউনিট জৱাহৰলাল নেহৰু চেণ্টাৰ অফ এডভান্স সাইণ্টিফিক ৰিচাৰ্চ বেংগলোৰত অৱস্থিত অনুষ্ঠানৰ ফেকাল্টি তেওঁ বিভিন্ন বিষয়ত সুন্দৰ গৱেষণা কৰি আছে এটা কেইবাজন ষ্টুডেণ্টৰ টিম লৈ তেওঁ কাৰেণ্ট ৰিচাৰ্চ ইণ্টাৰেষ্ট হৈছে উইক ইণ্টাৰেকশ্যন কনফৰ্মেশ্যন কণ্ট্ৰল hyperstable collagen peptides or peptidomimetics mimicking the peptide structure to solve various disease related and other disorders. So the title of today's talk is Vaccines and Drugs and Role of Academia. So we welcome Dr. Banikanta Sarma for this talk and we look forward to hear from you in this particular topic. Thank you. The stage is yours.
Thank you, Nilkumar. আৰু 15 মিনিট শিক্ষাৰ সকলোকে ধন্যবাদ জনাইছো এই অপৰচুনিটি দিয়াৰ কাৰণে so can I share the slide, Nilkumar?
Yes please
you can see, right? The slides you can see?
Not yet.
Okay, let me share it again. এটা দেখা পাইছে নেকি
yeah it's visible and আৰু আপুনি ফুল স্ক্ৰিনত কৰি দিয়ক
আচ্ছা
yeah
শুভ সন্ধ্যা আটাইকে আৰু ধন্যবাদ জনাইছো আকৌ এবাৰ আজিৰ মোৰ টপিকটো হ'ল ভ্যাকচিনছ ড্ৰাগছ এণ্ড ৰোল অফ একাডেমিয়া টপিকটো আৰম্ভ কৰাৰ আগতে মই অলপ ব্ৰিফলি কৈছো মোৰ গ্ৰুপৰ বিষয়ে যে এই টপিকটো একচুয়ালি যেটো টপিক মই আজি চুজ কৰিছো this is not my directly into my research area মই কাম কৰো হল peptides peptidomimetics related area যত মই weak interactions লৈ পেলাই কাম কৰো কিন্তু মই ভাবিলো যে যদি হে এটা মানে বিএসসি লেভেল বা এমএসসি লেভেলৰ টপ দিব লাগে তেতিয়া হলে মই ভাবো যে মোৰ টপিকতকে এনেকুৱা এটা টপিক লোৱাটো কিজানি ভাল হ'ব যেটো টপিক মানে আটাই ইন্টারেস্ট পায় মই নাজানো কেনেকুৱা হ'ব টপিকটো কিন্তু যেহেতু মোৰ এৰিয়াৰ নহয় এই টপিকটো I request all of you to take this topic as a general discussion topic এনেকুৱা নহয় যে তোমালোকে যিবিলাক ধৰণৰ কোৱেশ্চন সুধিবা মোক মই আটাৰে আনসাৰ দিব পাৰিম but I'll try my best. Okay, so মোৰ গ্ৰুপত বৰ্তমান I have 11 people excluding the summer students তাৰে 8টা PhD student PhD মানে কিছু in PhD student আছে যিবিলাক মানে in next 1 year or so they will enroll in the PhD program অসমৰ আছে 4-5 জন student so I am trying to maintain the male female and equal ratio so also I am trying to take students from all over India so if anybody is interested not only in my group in JNCS or IAC any group in India if you want to know about that group or you want to get some information always you can get in touch with me so I'll try my best to help you so as a topic to mostly I'll speak in English karon maximum jibilak word use kora hobo because it's related to drugs and vaccine ohomia shobdobilak moi najano okay so I'll be very honest with you so So let's get started with it. So before I get into this topic, let me wish all of you a very happy Independence Day, although it's belated and we are almost 1 week through. So I'll briefly discuss about India, because the Indian map and the human body has a lot of similarities. Okay, I'll just try to make a correlation. If you see, in India, we are surrounded by a lot of different countries, and towards the south, we have the Indian Ocean. So imagine a situation that some bad guys from any of these countries want to come in and invade India. So it could be a small terrorist organization, some, anybody you can consider for that matter who is not from India. So what is the first line of defense for us? The first line of defense is the border security forces, like the BSF. So imagine a situation that these bad guys overcome the BSF and could enter and mix with our general public. So what will be our second line of defense? So our second line of defense actually is not very proper. Okay, so once these people mix up with our general public, it is very difficult to detect them, identify them, and neutralize them. So imagine a situation that the government has developed a AI system, the artificial intelligence, in such a way that it can monitor our movement, each of us, 24/7, 360 days of the year. It's a bad situation. It will compromise our privacy and all. But the thing is, it has an upside that if any infiltration happens, the AI system can detect it and can inform us and help us to neutralize it. So why I'm giving this example is because our body exactly has this kind of a mass surveillance system. The surveillance system of our body is known as the immune system. So as I mentioned, that our body is very similar to this Indian map, that we have been all the time in contact with toxins, viruses, carcinogens, bacteria, etc. So the question is, if we are surrounded by all these things, why you are not sick all the time? What prevents us from getting sick? So what prevents us from getting sick is the philosophy based on which vaccines are developed. So I'll just discuss with you why we do not get sick and how vaccines are developed. I'll try to discuss this topic as kind of a story. You don't have to know the names of all the players involved in this process. Just listen to this as kind of a story and try to, correlate how vaccines are being developed. So if we look into our immune system, it's extremely complex. Scientists believe that the immune system is the second most complex system in our body after our brain. So there are two different ways how our body, reacts to the foreign pathogens or foreign materials. One is called the innate immunity, and the other is the adaptive immunity. I'll just briefly try to tell you what is innate immunity, what is adaptive immunity, how they are related with the production of vaccines. Okay. So immune system, innate immunity, adaptive immunity. Innate immunity is very non-specific. And this is called the first line of defense in our body. And adaptive immunity is the second line of immunity, and it is very specific to the bacteria, virus, or pathogen, whichever is entering our system. Okay, so innate immunity, innate as the word means, it's inborn. Okay, it comes when we are born. And it is non-specific in the sense that whether it is a virus or a bacteria or any kind of pathogen for that matter, this system reacts exactly the same way. Okay, so it doesn't have a specificity for the pathogen involved. And these two systems communicate with each other and protects us from foreign materials. So now the innate immunity system starts from our mechanical barriers like skin, the mucous membranes, the throat has mucous membrane, our nose has mucous membranes. Okay, so this membranes doesn't allow the pathogen to get into our body. Then, of course, we have also flushing mechanisms like, we have saliva, we have tears, urine mechanism. So all these flush out bacteria or viruses that get into our body. So not only they flush out, they also contains enzymes and all. So as you can see here in the next slide, we also have something called a chemical barrier. So chemical barrier in the sense that when we sweat, we produce a lot of antimicrobial peptides. So these antimicrobial peptides doesn't allow microbials to get into our body. So they repel this. Then we have this stomach HCl, which is called the gastric acid. So if you see its pH is 1, it means highly acidic. So what it means is that when we eat food, we not only take the food, we also take a lot of pathogen along with it, bacteria and all. And they need to be killed before the materials goes into our blood. Okay. So gastric acid plays the major role in this case, which kills a lot of bacteria because it has a very low pH. So similarly, we have enzymes called lysozymes in our tears and saliva, which also kills a lot of pathogens. So this is part of like initial defense mechanism, which is physical and chemical barriers, which is part of our innate immune system. So innate immune system also contains a lot of cells. Okay. So let us consider to discuss this cell, a situation where you have got a very small cut in your finger. So what happens, this situation you can consider as a invasion of pathogen. The moment you cut your finger, a lot of pathogens will enter your body through that cut. Okay, so now in such a scenario, to neutralize these invaders, we have a lot of cells that act as soldiers, captains, intelligent officers, suicide bombers, heavy weapons. We have several different kinds of cells. Okay, so the names of the cells I'll mention, but you can, like a story, you can consider them like soldiers, commanders or captains, heavy weapons, this way. You don't have to exactly know the name of the cells involved in this process. So basically the situation is something like this. Your bacteria or pathogens are invading you and your cells are getting ready. Okay, so it's kind of a, Kurukshetra war is starting. So the first cells in this process are those cells that are not killed during the cut. Okay, those are the cells, what they will do, they will scream and release some chemicals and inform the other cells that there is a cut and there are foreign particles or invaders coming in. So come and tackle the situation. So the first cells that will come to the cut are those called macrophages. And these are the soldiers that we have. What they do, they wrap around the bacteria or whatever pathogen you have and swallow them, digest them and kill them. So we have thousands of macrophages in our system. So one macrophage maybe can take up around 100 bacteria or viruses before they, get tired. So macrophages, once they get tired, what they do is they release chemicals and pass the information to something called neutrophils. Neutrophils are the suicide bombers. So they love killing. Okay, so the first thing what they do is they release a lot of toxic chemicals in our body. They don't care whether our own body gets harmed during the process, but their main job is to neutralize the invader. And they love so much the killing process that they sometimes commit suicide and produces a toxic net of chemicals. And this toxic net basically captures this bacteria or virus and kills them. So basically neutrophils love killing and they can even kill pathogens even after they die. So once this macrophages and neutrophils, they get tired, what they do is they again release chemicals and informs our body, which releases a lot of fluids. So our blood vessel releases a lot of fluids, and because of these fluids, you see a lot of time you'll see that your finger has swollen. Okay, so this fluid also brings something called complement proteins. These are some proteins that also love to kill bacteria and viruses. So this fluid, let me briefly mention about this fluid. So how they kill the bacteria or viruses. So this protein, they are large in number. There are about 30 of them, individual proteins. But if you calculate their numbers in our body, it comes to about 15 quintillions. So you can imagine that 15 quintillion killer proteins that are always floating around in our body. So what they do is they bind the pathogen and make holes inside these pathogens. So this is basically, this protein is inserting bombs inside pathogens. So what it does is, at one point of time, once this bacteria or the virus body is completely filled with this protein, they blast. So they basically, this is one of the ways. First is to cripple them and then rip through them. And another job of this protein is activating our immune system. So basically, they also, some part of this immune proteins also activate other cells in our body. So basically, what I have discussed here is the soldiers, the macrophages, the neutrophils, that are the suicide bombers, and the complement proteins. Okay. So once this battle gets intense and our body realizes that it may not be able to win this battle with the pathogen. Okay, if the pathogen is something like a coronavirus, you may not be able to win the battle with this cells itself. You need to activate your second line of defense. So these cells are basically part of the first line of defense. So what it does is, in this process, once we are almost losing the battle, these intelligent officers come into picture. The intelligent officers are basically the dendritic cells. So dendritic cells, what they do is, when our macrophages and neutrophils are fighting the bacteria, they collect information about this pathogen, that how this pathogen look like, what is the epitope or the peptide that this bacteria or virus has that causes diseases. So all this information it takes, and what it does is, it wraps its body with this pathogen and enters the superhighway of our body. The superhighway is the lymph nodes. So in the lymph nodes, we have the second line of defense, that are the B cells or beta cells. Okay, so, or bone marrow cells. So what it does, this dendritic cells, they get into this highway and look for some kind of cells. We call them as T cells or helper T cells. So what is the role of these helper T cells? So this helper T cells basically has a mechanism of detecting the virus or the bacteria that has invaded your body. So it's not any helper T cell. Our body has millions of helper T cell, but this dendritic cell has to find out a T cell that is specific for the pathogen that has invaded our body. For example, if it is a coronavirus, that helper T cell should be something that is specific to the coronavirus. Okay, so once this dendritic cell finds this particular T cell, it activates it. So now the helper T cell, which is also known as the commander or the captain, is now ready with weapons to fight the invader. So it is now getting activated. So now you may ask the question that how come our body has a cell that contains the weapons against a virus or a bacteria that we have never seen? Okay, so the simple answer to this question is that our immune system has cells that has got weapons against any disease that is existing in this planet or the universe, or that will come in next 100 years. Okay, our body has a mechanism to produce this kind of cells. Okay, so once these T cells are recognized, what they will do is they will multiply themselves. Okay, they will multiply in numbers and thousands of them will be produced, and half of them will go to the battlefield and they will help the macrophages. So the macrophages, they were tired and almost dying, So what these T cells will do is they will go and whisper something in their ears and it will activate them. You have seen a lot of Bollywood movies. So Bollywood movies, you will see that the hero or the side hero in this kind of war movies, they will try to inspire our soldiers, okay, as a last attempt to neutralize the enemies. So same function is done by the helper T cells here. They try to inspire the macrophages, which get activated again and fight a last battle. Okay, the second set of helper T cell, what they will do, they will move through the fluid in the lymph nodes and find something called B cells. Okay, so what are these B cells? B cells basically are the heavy weapons I was discussing about. So B cells are cells that are produced in the bone marrow. So that's why they are called B cells, and they produce a lot of antibodies. So B cells also possesses these antibodies on its surface. Okay, so now the T cell doesn't only find out a B cell, it also finds out a specific B cell for that particular pathogen. So basically, if again coming back to the coronavirus example, if it is a coronavirus that has invaded our body, the T cell has to find out a B cell that is extremely specific for the particular coronavirus. Okay, so what it does, it recognizes the coronavirus by using these antibodies. Antibodies, you can see it has hooks which binds to the particular regions of bacteria or viruses. In this particular case, it will bind to the spike proteins of coronavirus using these hooks. So I'll briefly mention about in a subsequent slide how these hooks, what are these hooks and how they bind. Okay, so basically, as I mentioned in the previous slide, that we have our body has a system in such a way that it can detect disease via or detect pathogens via T cells. Okay, similarly, our body also has a B cell, okay, that can actually find out or bind to any pathogen that is known in this universe, or a pathogen that will be produced after 100 years in this universe. So our body has a B cell. So once the T cell finds the B cell multiplies. Okay, so once they coordinate with each other, B cell multiplies, and B cell multiplies, not only it multiplies, it also starts producing these antibodies. So each B cell can pump around 2000 antibodies per second. So you can imagine how much antibodies the B cell produce during the day once B cells are activated. One of the major issue here is that the B cell activation takes a lot of time. So I'll come to that point, why we need vaccines and all. If we have such a strong defense mechanism, why you need vaccine. So this subsequent slide, this is the most important slide on this presentation. Okay, so here I'm going to discuss how this immune response and our vaccine production is related. Okay, so the antibodies, how they kill pathogens, they again binds this pathogen. As you can see, the antibodies are produced like 2000 antibodies per second. Billions of antibodies are produced in the body, and they wrap around the pathogen and make sure that they get immobilized. They cannot move. Okay, once they binds to this pathogen and make them stationary, the other cells comes into picture and starts killing this pathogen again. Okay, there is also a mechanism which is called cell-mediated mechanism involving killer T cells, which I'm not going to discuss here. So this is called humoral mechanism. There is also a T cell-based mechanism by which pathogens are killed in the second stage of our defense mechanism. So basically, once this antibodies binds to this pathogen, the macrophages and neutrophils clears the battlefield. What happens is that our body now knows that pathogens are neutralized and there is no need of neutrophils and macrophages, and they start killing themselves. But all of them doesn't kill. Okay, so what happens is that here the helper T cell that recognize this pathogen, okay, some of them converts to memory T cell, and the B cells which also recognize this pathogen also convert some of them to memory B cells. So this memory is very important in the sense that, so suppose now this pathogen got neutralized, but this pathogen again can attack us after suppose 6 months, 1 year, 2 years. Okay, so in that case, what will happen is that this memory T cell and memory B cell whose activation took a lot of time in the first invasion by the pathogen, will now be already present in our blood. Okay, and only thing they have to do is they have to multiply themselves. So our response to this pathogen during the second attack will be extremely fast. Okay, so this is the basis of basically producing vaccines. So what vaccines does is, vaccines activates our system in such a way that it produces memory T cells and memory B cells. So vaccine is basically, if I say in terms of cricket, suppose Indian team is going to play in Australia. So before playing their first test in Perth, what they will do is they will play some warm-up games. Warm-up games with teams that are relatively weaker compared to the main Australian team, so that they get used to conditions. Okay, so same way we can make our body get used to conditions of pathogen invasion. So basically, you can say that vaccine is basically practice match before the actual pathogen or the bacteria or the virus invades our body. So the vaccine will not produce the disease, or the full-blown disease will not be produced by injection of the vaccine, but it will produce the T cells and the B cells that are required to fight this pathogen. Okay, so we basically can activate our immune system by using these vaccines. So now, to sum up what I have discussed so far, so you had a cut. After 2 weeks, maybe you will see some red patches in your finger. So after 1 month, you will see that you basically don't see anything. Your disease or your cut is completely healed. So your body has gone through a Kurukshetra war, but you do not know about what was happening in your cut. Okay, so that's how our immune system works. So now, to summarize the effect of our immune response, we have two mechanisms. One is the innate mechanism, which is inborn. Okay, the other is the adaptive immunity. So say here I want to mention one point there. So why we need vaccines for kids or the babies? So this is because our innate mechanism is already in place when the child was born, but our adaptive immunity takes time to develop. So that is why vaccines are given to kids, so that they can develop this adaptive immunity. Okay, so that their B cells or T cells are produced against this common, pathogens like influenza virus, typhoid, all these things. You can always get vaccinated and get your body ready with antibodies and T cells to fight back. So now coming back to, again, innate immunity and adaptive immunity. Innate immunity is our first line of defense, and adaptive immunity is our second line of defense. So innate immunity has no time lag, meaning the moment the pathogen attacks you, this innate immunity is ready to fight. Okay, and it is not antigen specific, so it's basically common to all pathogens. Whatever pathogen it is, it will act the same way. Okay, and it doesn't have a memory. So next time when this pathogen again attacks you, your innate immunity doesn't recognize the pathogen. Okay, but adaptive immunity, because it is via T cell and B cell, it has a lag period. Okay, lag period means it takes almost 1 to 2 weeks to develop the antibodies in your body. Okay, same thing happens when you are vaccinated. Production of antibodies takes 1 to 2 weeks. Okay, so, and it is antigen specific. That means if it is a coronavirus as the pathogen, you will have a very specific T cell or B cell against this antibody, and there is a memory associated with it. So it's the second time if this same virus attacks you, your adaptive immunity can recognize it and produce the neutralize it via the antibodies and the T cells. So now, as I said before, that I'll briefly mention how these antibodies look like, because this antibody is the key in our vaccine production and all. So antibodies, as you have seen, this is the B cell, pictorial representation of B cell. It can have two forms. One is the soluble form, which is shown here with these dots, and other one is the membrane bound form. Okay, B cell, and these are also known as B cell receptors. Okay, so antibodies can be in either form, either in soluble form and the membrane-bound form. Okay, so as I also mentioned, it's a Y-shaped molecule. So this molecule basically is something like this. So it's a polypeptide, long chain peptide with hundreds of amino acid residues. Okay, so it has a Y shape and it contains two chains of peptides. One is called the heavy chain, the other one is called the light chain. Okay, so basically this Y shape, this Y portion is specific for the antigen. So this is the portion shown in red here. This is the variable portion of the antibody. And this blue ones are the constant region of the antibody. It is called the Fc region through which this antibody binds to the B cell. Okay, this is more or less constant for all the antibodies. But the antibody differs in this portion, this highlighted in red here, and this portion is the antigen specific binding site. So antigen means it could be a peptide or a polysaccharide, okay, coming from the this is specific pathogen. For example, in case of coronavirus, it is called the spike protein, which is basically is the antigen that binds to the antibody. Okay, so now if we have such a good immune system, okay, that body produces a lot of antibodies, T cells and all, and fight back with pathogen, why do you need vaccines? Okay, this is because the body needs time, okay, a week or more to produce antibodies against the pathogen. Okay, suppose coronavirus infects someone, by the time the antibodies come and fight back, it is around 10 days or a week or two. So by the time, if the pathogen is severe, the person can die. Okay, so we cannot take the risk of this, that antibodies will be produced naturally by this, pathogen. If body is weak or young, we are always at risk. Okay, so that is why we need to trigger our immune system with a milder version of the pathogen. That's what I was talking about the practice match. We have to do some practice match and produce antibodies and make our body ready for the fight. Okay, so the way to do it is to get vaccinated. Okay, so now how are vaccines produced? So there are multiple ways in which we can produce vaccines. So the oldest one is something called live attenuated vaccine. So which means is that this is a live pathogen. Okay, if we're talking about coronavirus, it's the live coronavirus, but a weakened version. Okay, it's a laboratory produced coronavirus which is much weaker than the one infecting people all around. Okay, so what it does is it produces a immune response in our body. But the problem with this live attenuated vaccination is that if your body is weak, you may not be able to tolerate this kind of vaccines because the virus is live. And if something goes wrong, the virus may replicate in your body. Okay, so normally this kind of vaccination is not preferred. Okay, so the other way of doing it is using an inactive vaccine. Inactive vaccine means you use a dead pathogen. That means you use the coronavirus, but before you use it, kill it. By using heat, radiation, whatever way, chemicals, you can kill it. So as you can see here, if your danger level of strong pathogen is this, the dead pathogen has a danger level of which is much reduced. But the advantage is that although the pathogen is dead, all the materials or genetic materials that this pathogen had, okay, all are intact. The pathogen cannot replicate in your body, but whatever proteins, peptides, polysaccharides, DNA this pathogen had, all are injected in your body along with this vaccine. So the Covaxin that is being used for coronavirus produced by our Bharat Biotech is a inactive vaccine, which is a dead coronavirus variant. Okay. The third variation is something called subunit vaccine. Subunit vaccine is, it is not going to inject you with the pathogen or the dead pathogen, but what it will do is, it will take a portion of the bacteria or virus and produce a vaccine. So in case of coronavirus, this is the surface protein. This is the spike protein I have shown in red here. This is the spike protein that we can use to create a vaccine. Okay, so Novavax vaccine that is used now for coronavirus is a peptide-based or spike protein-based subunit vaccine that are being used. Okay, the first two cases, as you can see, are good in the sense that it will have all the genetic material of the virus or the bacteria. Although it will not produce the full-blown disease and pathogen cannot replicate, but it will produce a strong immune response in the sense that all the T cells and B cells that are required to fight back this pathogen will be activated in your body. But subunit vaccine, because it is being used with a very small portion of the virus or the bacteria, it is sometimes not very effective. Okay, not very effective in the sense that you have to use something called adjuvant along with these vaccines. And it may not create a long-lasting immunity, and you may need to take booster dose. Okay. So now coming to some of these more modern approaches, which are based on nucleic acid approaches, DNA and RNA vaccines that are being produced now. Okay. So what in these approaches are being done is, you are not going to inject your body with the peptide or the virus itself. But what you will do is take a portion of the DNA of this particular virus, okay, and inject this DNA in such a way that the DNA can undergo transcription to messenger RNA, and the messenger RNA can produce the peptide or the protein responsible for the disease that is being caused by the pathogen. For coming back to again coronavirus, let me tell you, if you inject the coronavirus DNA, what it will do is it will produce the messenger RNA. The messenger RNA in the cytoplasm will undergo translation and produce the spike protein. And what this cell will do is, this is our human cell, okay? The cell has a mechanism of taking this protein outside its surface with something called major histocompatibility complex, okay, MHC1. So it will bind the protein, and the protein will now be produced in the surface of the cell. And now other part of the body will know that, okay, our body has a foreign material that is being shown by our cells as a surface protein. So now against this surface protein that is being produced inside our own cell, our body will produce antibodies and T cells. Okay, our body is very sensitive to any foreign materials, okay, because this peptide is produced inside our cells, but this peptide is not part of our genome. Okay, then our body can recognize this as a foreign material. So our body is so sensitive in recognizing foreign materials that sometimes it attacks our own body parts. You may have heard about this name, autoimmune diseases, where our body or our T cells and B cells attacks our own cells, okay, or our own proteins. Okay, so now coming to one of the common problems with this DNA vaccine is the insertion of this DNA inside our nucleus. It is not that easy. Okay, and the other problem is that if you insert a viral DNA inside our own DNA, it can integrate with our DNA system. This risk is always there, although scientists say that this should not happen or doesn't happen, but the risk is always there that what if this viral DNA integrates with our DNA systems? Okay, so one of the ways to insert this DNA inside our body cell is by doing something called viral vector design. So basically, you can take a virus that is not maybe human origin, okay, something like this Covishield that are being produced by AstraZeneca, is a viral vector DNA. What they do is they take a chimpanzee virus, okay, chimpanzee virus they take, but it removes chimpanzee virus DNA and inserts the coronavirus DNA inside it, and let this virus go inside our cell. So basically, this cell acts as a medium to take this DNA inside our body, okay, to the nucleus, from the cell to the nucleus, because DNA once you inject a DNA in our body will recognize it as a foreign particle and produce immune response and destroy the DNA. So before that happens, you have to insert this DNA inside the nucleus. So this is done by using a cell. Okay. So this viral vector DNA is very common now, and several companies are working towards producing viral vector DNAs, because in this case you do not have to insert the pathogen or the spike protein inside your body. Your body can itself produce the spike protein against which T cells and B cells can be produced. Okay, and the final one is something called mRNA vaccine. So you do not even have to insert the DNA in your body. You just put the messenger RNA of coronavirus inside your cell cytoplasm. It doesn't have to go to the nucleus. And now, because you do not insert the DNA, there is no question of integration of viral DNA with human DNA. So this messenger RNA can produce the antigen which will be produced in the surface, and our body can recognize it. Okay. So now, to cut this long story short, what we do is we inject any form of vaccine, okay, whether it is a full-blown pathogen vaccine, deactivated pathogen vaccine, peptide vaccine, or DNA vaccines. What it does is it primes your body or makes your body ready for the fight by producing T cells and B cells, the second stage of immune response. It triggers a second stage of immune response and makes your body ready for the fight. So that's what it does. So now, many times I hear this question, how vaccines are different from drugs and all. So why people can produce like the coronavirus vaccine in a year or two, but they cannot produce drugs in 1 year or 2 years. This is because, see, when you are, that you are being attacked by coronavirus. So if we can produce the coronavirus in lab in a short span of time, we can, identify all the information about this virus, and it becomes easier to produce vaccines. But for diseases, most of the time, what happens, we do not know the root cause. Identification of the root cause takes a lot of time in case of drug development. Okay, so what I'll try to discuss in the next part of my talk is I'll briefly discuss about how drugs discovery works, okay, why it takes so much time and why drugs are expensive, and what kind of role and the academic setup can have in this kind of vaccine or drug production. So to know how drug works, okay, we should also know something called our digestive system. Okay, our digestive system is the third most complex system in our body after brain and the immune system. Okay, so digestive system mechanism we should know, because you have various types of drugs, some oral drugs, some drugs are injected. So some drugs are given as patches that go into our body through skin. Okay, so why this kind of mechanisms are necessary, and why some drugs can be given as only injection, not as oral tablets and all this. A lot of different things are there in the discovery process. I'll not go into the details, but I'll just try to briefly mention how drug discovery differ from vaccine discovery, and how drugs are different from vaccines. So vaccines are basically, as I discussed, they are not drugs. They make our own body ready for a fight. But drugs are something that kill the pathogen themselves. Okay, for example, if you take antibiotics, our antibiotics doesn't make our body ready for a fight back, but it kills the bacteria by itself. Okay, so drug has a specific purpose. Okay, and vaccine has a single purpose, that is producing our immune response and make our body ready for the fight. Okay, so coming to the digestive system, it starts with our mouth. Okay, when we eat something, we swallow it, it passes through the esophagus to our stomach. Okay, and then through stomach, it goes to the small intestine, and then it goes to the large intestine, and then it goes out from our body. Okay, so this is the most simplistic way of explaining. Then from the small intestine, once the food is being digested in this stomach, it comes to the small intestine. It comes in the form of small monomers as well as oligomers. For example, if I say monomers, if you take carbohydrate, okay, sugar or glucose is the monomer. If I talk about protein, if you eat meat, protein, the monomer is the amino acid, and so on. So the materials that reach from the stomach to the small intestine could be either of them. It can be the amino acids and the glucose, or disaccharide, triglycerides, and polypeptides and all. So the small monomers that can pass from this small intestine to our bloodstream through a lining between the bloodstream and the small intestine. Okay, but the bigger particles or the bigger dimers, trimers, oligomers, they cannot pass from this small intestine lining to our bloodstream. Okay, so what they will do, they will go from small intestine to our liver. Okay, and liver will process it. So liver is the most important organ in our body. It works as a factory, storage house, processing unit, as well as distribution unit. So it does all the things. It takes blood from heart as well as intestine and purify it and distributes in our whole body. So second phase of monomers like glucose and amino acid reaches our body from our liver. First passage is through intestinal wall, second is from liver. So now any drug that you take, orally take, it has to go through this whole process. It has to go from your mouth all the way to stomach and pass through intestine as well as go to liver. And this process is lengthy and takes many, many hours. Okay, most of the drugs are not stable under these conditions. As I mentioned earlier, that our stomach has gastric acid that is around pH 1. It destroys all the peptides in that low pH. So peptidic drug, if you're working with, you cannot basically swallow. You have to maybe give it through injection or like a patch. Okay, so that it directly reaches your bloodstream without going through this process of, stomach and all these things. But still, our liver will destroy most of the peptides that we inject in our body. Okay, only a small amount of the drug that we take can pass through liver to our bloodstream. See, unless something goes to our bloodstream, the drug is not effective. It has to go to our blood, then it has to go to our cells. Okay, so here I'll briefly discuss about diabetes, insulin response, and what kind of drugs are being produced for diabetes. So diabetes I have taken because you see, if you look around, any male in Assam or all over India, 30 plus, 40 plus means you will see two things: a big belly and sugar problem. So we should know how the sugar metabolism works in our body and what kind of precautions we should be taking, to get rid of diabetes. Okay, so diabetes, one of the important, molecule in diabetes is this peptide called insulin. It's a hormone. See, you hear a lot of names, hormones, antibodies, this and that, all are peptides. Okay, the name should not scare you, because if you look into this is a hormone. Its function is a hormonal function, but it's a peptide. As you can see here, I have given you the amino acid sequence there. It has two chains, A and B, and they are bound by disulfide bonds. So let me briefly discuss what insulin does and what happens to our blood sugar in presence and absence of insulin. So we have two kinds of, diabetes, type 1 and type 2. If you do not know, like type 1 diabetes, let me briefly mention, I am also not an expert in this area, but I'll just try to give you just gist. Okay, type 1 diabetes means see, I have shown you here a very small organ. It's called pancreas. Pancreas produces beta cells. Okay, this beta cells produces insulin. So in people who are suffering from type 1 diabetes, they mostly do not, this pancreas do not produce beta cells. That means they do not produce insulin. And this type 1 diabetes is often seen in very young age, like people in the age below 20 years. Okay, mostly this disease is seen in those people. Okay, child, maybe 12 years, 13 years, 14 years child, you will see that mostly they are having this problem of type 1 diabetes. That means their body is not producing any insulin. And the only medication they can have is insulin shots. Okay. The type 2 diabetes, your beta cells are producing insulin, but somehow your body is not able to use this insulin, or maybe your body is not producing enough insulin to metabolize all the glucose. So now how this insulin and glucose are correlated. Let's look into this. See, you can see this oval shaped red line I have drawn here. You can consider this is a human cell. Okay, this is inside the cell, this is outside the cell. As I mentioned here, when you take food, it can go from esophagus to stomach to small intestine, and glucose from small intestine can be released to our blood circulation. So now once it reaches our blood circulation, what happens? It is outside the cell. This glucose that is outside the cell is useless until and unless it can go inside the body, inside the cell. Once it reaches inside the cell, this glucose has to be degraded to ATPs and all, and then only our body can produce energy. So glucose basically goes to ATP, carbon dioxide, water, and energy is released in our body. Okay. So now, unless this glucose can go inside our body, inside our cell, it is going to accumulate in the bloodstream. It is useless and it will create high blood sugar level. Okay. So now how this glucose molecule can go inside our cells? The glucose can only go inside our cell only when insulin binds something called insulin receptor. So pancreas will produce insulin. Once glucose will be there in our bloodstream, our body will respond in a way that beta cells will be triggered to produce more insulin. And insulin will come and bind to this insulin receptor. So it's kind of a key which will unlock this receptor through which glucose can go inside. So insulin is the key which will open the gate for your blood sugar to go inside the cell. Okay, unless insulin can bind here, it cannot go. Okay, so once glucose goes inside, it can produce energy, it can trigger a lot of different processes. Glucose can be converted to glycogen, which can be later on broken down to produce more glucose once body needs energy. So glucose is the main source of energy, but it is useless in the bloodstream. It can produce energy only when it goes inside the cell. And it can only go inside the cell only when insulin binds to the insulin receptor. And insulin receptor, if people having conditions where insulin receptor is not sensitive to insulin, then there is a problem. And if insulin is not being produced in your beta cell, then you need to take insulin shot. But insulin binding to the receptor is essential for glucose metabolism. And glucose can come inside the cell only when insulin binds to the insulin receptor. So there are also other players in this mechanism, something called glucagon that are produced by the alpha cells in the pancreas. So I'm not going into those things, but the key thing here is insulin has to bind to our cell for glucose to enter our cell. Okay, insulin is the key. If it opens the gate, then only glucose can come inside our body. So now, how to tackle this problem of, insulin and glucose, high blood sugar and all. There are a lot of different kinds of proteins involved in this pathway. Okay, so as I was telling, if your body is not producing enough insulin, then what you can do? You can activate your beta cells. Okay, that will produce more insulin. So there is a type of drugs that are known to activate our beta cells. Okay, so then there are kind of drugs which, as I was telling, your insulin can bind here. Maybe your receptor is not sensitive towards insulin. So what you can do is you make your receptor more sensitive to insulin. So there are kind of drugs that can make, your body more sensitive to insulin. So, for example, there is a famous drug called metformin, which makes our insulin receptors more sensitive towards insulin. So same way, we have medicine that what it can do is it can tell our body that, okay, you digest your carbohydrates slowly so that you produce less and less glucose. Okay, so there are drugs that slows down the metabolism of our carbohydrates. You know, there is a famous category of drug called alpha glucosidase inhibitors. Okay, so there are different kinds of mechanism, different kinds of proteins we can inhibit in this process. So basically, for tackling diabetes, we do not have a single pathway to produce drugs. So this is what makes life difficult. You do not know for a person which drug will work. Okay, which stage of diabetes he is in, whether more production of insulin will help him, whether sensitizing insulin receptor will help him, whether slowing down the carbohydrate will help him, or if nothing works, there is only one way, then you have to get insulin shots, injections of insulin. Okay, so you see, in vaccines, we have only single way of doing it. You inject a vaccine, sensitize your body and prime your cells and antibodies so that they can fight back. But for diseases, it's much more complex, and we have to know exactly which pathway to trigger to cure this disease. Okay, so it's much more complex in case of diseases compared to vaccines, and that's why it takes a hell lot of time to produce drugs. So I'll briefly mention here how this drug discovery process is done. So as you can see here, I have shown different stages of drug discovery. Okay, so first step is target identification. So what target identification means? I mentioned that the activation of, suppose, beta cells to produce more insulin. But there are maybe 100 proteins in beta cells. We have to know which proteins we have to target to activate the beta cells. So that itself takes maybe 5 to 10 years of research to identify the target. So once you identify the target, that okay, if I hit protein A, it activates the beta cells to produce more insulin. Now you have to develop an assay. Okay, develop an assay with the purified protein, develop an assay with the protein in cells. So then develop an assay for the protein in animal models. Okay, there are a lot of works involved in that. Now, once you know that this is the target, protein A is the target, what you have to do is you have to make a molecule that can activate this protein. Okay, so that is something called lead identification or hit identification. So once you identify a molecule that can bind protein A to activate beta cells, now your initial stages you will get something, a low affinity ligand or low potency ligand. Potency means its performance is less. So now you have to optimize it. So optimization of this ligand can take you a few years. Okay, so once you optimize this hit and you get a lead, so what you have to do now is you have to do some preclinical studies. Preclinical studies means you have a very good molecule that activate protein A in in your body, in purified state or in a cell line. But now you have to see how this molecule works in an animal model, for example, in a mouse model. So you have to do a lot of works with animals and identify that this protein really triggers beta cells and establish that this protein is responsible for your beta cell activation and insulin production. So then you reach the phase 1, phase 2, phase 3 clinical trials, and this whole process takes you around 10 to 17 years. And the cost is around overall $1 to $2 billion. Okay, so as you can see, it's a very long process and it costs you a lot. So that is why our drugs are not that cheap. Your vaccine maybe government can give for free, but not the drugs. Okay, because until and unless the generic drugs comes into picture, it is going to be expensive. So let me also briefly tell you what is generic drug, what is branded drug. So generic drug are more or less same as the branded drug. The brand is, for example, suppose the Pfizer, GlaxoSmithKline, or any company for example, makes a drug, they will have a patent. Okay, it will last for around 15 to 20 years. So what it means is that for 15 to 20 years, no other company can make the same drug. Once their patent expires only, then others can make the drug. So when others make the drug, it becomes cheaper. Okay, the others make the same drug, but their formulations can be different. So basically, the drug, there is something called API, called active pharmaceutical ingredient. Okay, that means it's the molecule that is active. But along with that, you have a lot of other things also added, like adjuvant, something that helps in stabilizing your molecule in cells, stabilizers, and a lot of other things are used along with the active pharmaceutical ingredient to produce the drug. So that formulations can be different from company to company, but API will be same. So now once this generic drug comes, mostly our Indian, pharmaceutical companies become active and it becomes cheaper. The drugs become cheaper. Okay. So now, so this is the same overall process I'm talking about. So when you want to discover a molecule against a particular target, what you do is you screen a lot of compounds. Here I have given you a very small number, 10,000. This number goes from 10,000 to 100,000. Okay. You test this much molecules, and in preclinical phase, you have only maybe 250 to 500 compounds you identify that may likely to go forward for the clinical stages. And then by the time you come to clinical trials, so 3-4 compounds will last. And then by the end of your 20 years, you'll see that only one compound you have identified that can be used as a drug. Okay, so basically you are coming from a lakh or 10,000 compound to 1 or 2 compound that can be used as a drug. And now, how to screen this many molecules? How to identify that from 1 lakh molecule, how to identify a maybe 50 molecules or 100 molecules that are active. So what they do, there is a process called high throughput screening. So you use a robot to do the manual works. Whatever small works we do, assays and all in our labs, in academic lab, this is done by robot here. Because 100,000 compound screening is not possible. My whole career will be gone, maybe screening, 10,000 compounds. But if you want to screen 100,000 compounds in a very short span of time, you need something called a robot, which can do the assays for you. Okay, the human work can be cut down by a robot, and it will be very, very efficient. As you can see, these robotic systems and all are not possible in academic setup. This is only possible in mostly pharmaceutical industries. Okay, that too also, if you think about this kind of a screening system, this may be possible only in a very, advanced labs. Okay, pharmaceutical labs also will not have this kind of facilities. At least in India, I don't know of any labs who does this kind of very big high throughput screens. Okay, so now target identification, hit generation, lead identification, then post approval and all. So all these things can be done in academic setup. A biologist can identify a target in biology lab. A chemist can identify a hit. Okay, basically the molecules, that 1,10,000 molecules you are screening, they're not produced directly in the pharmaceutical industries. They borrow this molecule from different academic labs. So there is a mechanism of collecting, new molecules all the time from the academic labs, and they are being stored in pharmaceutical industries. Okay, so molecules, production of this molecules can be done in academic setup. Target identification can be done in academic setup. Even lead generation can also be done in academic setup. But the problem comes when we have to do, preclinical studies and clinical trials. The academic setup cannot do this kind of studies. Okay, so let me briefly tell you what are these, clinical trial means. A lot of times we confuse clinical trials. See, both vaccination and drug discovery have clinical trials. In vaccination, clinical trials are always involving healthy humans. Okay, because you are trying a vaccine, you cannot start with a coronavirus patient. Okay, you have to start with a healthy volunteer. But in drug discovery, there are multiple phases where you can use healthy humans as well as sick person. Okay, so the first stage of clinical trial is basically by using a set of healthy humans. When in phase 1 clinical trial. So basically what you do is you administer this drug in a small number of healthy volunteers. Okay, these are basically the guinea pigs. I don't know whether advanced countries you'll find any volunteers for this kind of injections, but mostly I think African people are being used in this kind of first stage of clinical trials. So what basically this phase does is, this phase doesn't look into the efficacy of the drug, because you cannot look for the efficacy because the person doesn't have the disease. So what it does is it sees the effect of the drug on our body and the effect of our body on the drug. And you might have heard these words, pharmacokinetics and pharmacodynamics. So pharmacodynamics means what the drug does to you, and pharmacokinetic means what your body does to the drug. Because as I mentioned, your body will have an immune response against this drug because it's a foreign particle. Okay, whether your drug molecule that is being injected in your body, whether its half-life is 30 minutes, 1 hour, 1 day, 1 week, you have to analyze all these things, which will determine the dose of this drug. Okay, in the second stage, what is done is, you take some small number of persons who are suffering from the particular disease against which you are producing this drug. So in this case, you check both safety as well as efficacy of the drug. Okay, safety, because in the first stage you have checked safety in healthy humans. Now you have to check safety in patients. Okay, and then now also you will see efficacy. And third stage is also basically similar to second stage. Phase 2 and phase 3 clinical trials are similar, but the number of people being tested drastically increases in phase 3. If you are doing phase 2 in 200 people, phase 3 maybe you are doing in 2,000 people. Okay. Also in phase 3, you look into different kinds of data, like how this drug behaves in younger generation, how this drug behaves in women, how this drug behaves in men, how this drug behaves in old people, how this drug behaves in Indian population versus Chinese population. All this data are being collected in phase 3 clinical trial. Once the phase 3 clinical trial is over, you submit this molecule for approval from FDA or whatever body that this particular country has, and you get an approval. Okay, once the approval is obtained, you have this phase 4 clinical trial going on. That means the company who has produced this drug is still monitoring the effect of this molecule in different populations. Now, because this molecule is mass produced and now distributed in different countries, now you can get a lot of data regarding this drug in different kinds of populations. So this company keeps monitoring this data up to 4 or 5 years. Okay, so basically this process is a very lengthy process. As you can see, it takes up to 10 to 20 years, and it's very, very expensive. And this is why our drugs are basically relatively much more costly compared to vaccines. Okay. So, yeah. So these things I have already discussed. So now let me briefly talk about what I have been telling you about the role of academia. So as I mentioned, see, a lot of stages of the drug discovery we cannot do in our academic setup. Like clinical trials, basically we cannot do. Animal studies to certain extents, many labs can do, advanced labs, but not all kinds of lab can do, animal model studies. But the thing is, the academic setup can help in identification of targets. Like we can help in discovering a protein that could be useful for targeting a particular kind of disease. Okay, whether it is a cancer, diabetes, or pneumonia, whatever. If a disease is happening, we can identify a particular protein or an enzyme that is responsible for the disease, and we can also help produce molecules that can bind to this protein, neutralize this protein. So chemists can do this, biologists can do the target identification stage. And also nowadays, a lot of companies are not interested in producing antibiotics and drugs that are not a long-term, that doesn't have long-term prospect. For example, if you have something like bacterial infection, you take antibiotic, you are cured in 10 days. So the pharmaceutical company is not getting much benefited by an antibiotic. But suppose you have diabetes, you will take that medicine throughout your life. So the pharmaceutical company is more interested in producing a drug for diabetes than antibiotics. So with government funding and charitable and philanthropic organization funding, lot of academic labs, at least in the advanced countries, have started this drug discovery campaigns, at least for those diseases that are being neglected by the pharmaceutical industries. Okay, so yeah, so now coming to, this is basically a table where I have tried to summarize some of the drugs that have academic origin, starting from the Taxol that used for cancer therapy. So there are many drugs whose origin have started in academic labs. For example, some academic lab has got a compound that binds to a protein that is important for cancer biology. So that molecule, the pharmaceutical industries can take forward in developing drugs, even if not exactly the same molecule. After medchem and lead optimization, they can come up with a better molecule which structurally resembles the molecule that academic lab has discovered, okay, and they can take forward. So in a nutshell, what I want to say is that although our academic labs cannot produce drugs, they can in some way or the other help the pharmaceutical industries in producing drugs. Okay. So with this, I would like to conclude that research on vaccine and drug discovery is very interesting, challenging, and rewarding. Okay, researchers with expertise in both chemistry and biology can significantly contribute to this area, like drug discovery. And academic drug discovery, is gaining popularity and momentum. And in many advanced countries, in many advanced institutes, for example, Cambridge, Harvard, they have now started drug discovery programs in academic setup. Okay. With this, I would like to thank you all for your kind attention. I would be happy to take any questions that you might have. Thank you.
15 মিনিট শিক্ষাৰ তৰফৰ পৰা ডক্টৰ বাণীকান্ত শৰ্মাক আমি অশেষ ধন্যবাদ জনাইছো। ঘণ্টাতকৈ বেছি সময় কাল তেওঁ ইমান সুন্দৰকৈ আমাৰ যেটো জন্মগতভাৱে পোৱা প্ৰতিৰক্ষা ব্যৱস্থা, দেহত থকা প্ৰতিৰক্ষা ব্যৱস্থা বা পৰিবেশৰ লগত মিলাই বিকাশ হোৱা যেটো প্ৰতিৰক্ষা ব্যৱস্থা, হেতু এখন দেশৰ প্ৰতিৰক্ষা ব্যৱস্থাৰ উপমাৰে ইমান সুন্দৰকৈ আমাক বুজাই দিলে। তাৰ পিছত ভ্যাকচিন আৰু প্ৰতিষেধক আৰু দৰবৰ ৰোলটো কি বা ভূমিকা কি হে সম্পৰ্কেও তেওঁ ইমান সুন্দৰকৈ আমাক কথাখিনি, এইটো আসলতে হাতিমাৰি ভৰুকাত ভৰোৱাৰ নিচিনা হৈছে, কাৰণ ইমান ক'ব লগা কথা আছে। এই কথাবিলাক অথচ ইমান সুন্দৰকে আটাইখিনি গুৰুত্বপূৰ্ণ দিশ আমাক কোৱাৰ বাবে, আমাৰ ছাত্ৰ ছাত্ৰীসকলৰ বাবে তেওঁ দাঙি ধৰাৰ কাৰণে তেখেতক অশেষ ধন্যবাদ জনাইছো আমাৰ 15 মিনিট শিক্ষাৰ তৰফৰ পৰা। আমি প্ৰশ্নোত্তৰ শিতানলৈ যোৱাৰ আগতে আমাৰ পৰৱৰ্তী অনুষ্ঠান সম্পৰ্কে এটা ঘোষণা দিব খুজিছো যে আমি আমাৰ যেটো পৰৱৰ্তী অনুষ্ঠান, এই পৰৱৰ্তী অনুষ্ঠানটো হ'ব সেপ্টেম্বৰৰ 4 তাৰিখ, আৰু 4 তাৰিখে আমাৰ অতিথি হিচাপে উপস্থিত থাকিব পুষ্পধৰ দাস। পুষ্পধৰ দাস অসমত বিশেষকৈ এতিয়া এজন অতি গুৰুত্বপূৰ্ণ লোক এইফালৰ পৰাই, যে তেওঁ সোণাপুৰ হায়াৰ সেকেন্ডাৰীত পঢ়া শুনা কৰিছিল, তাৰ পিছত তেওঁ সেন্ট ষ্টিফেন্সত পদাৰ্থ বিজ্ঞান পঢ়িছিল, তাৰ পিছত IIM বেংগালুৰুত তেওঁ MBA কৰি অসমলৈ ঘূৰি আহি অসমৰ বাক্সা জিলাত খেতি কৰে, তেওঁ খেতিয়ক। গতিকে তেওঁৰ যেটো যাত্ৰা, যেটো বৰ্ণিল যাত্ৰা, আৰু তেওঁ যিবিলাক কামত এতিয়া কাম লৈ এতিয়া ব্যস্ত হৈ আছে অসমত, কৃষিৰ ক্ষেত্ৰতেই হওক বা দুগ্ধ, আমাৰ যেটো দুগ্ধ উৎপাদনৰ লগতো তেওঁ ওতঃপ্ৰোতভাৱে জড়িত। গতিকে তেওঁৰ এই যাত্ৰা, তেওঁ দেখা পোৱা সুবিধাবিলাক, তেওঁ দেখা পোৱা অসুবিধাবিলাক, আৰু কেনেকে শিক্ষা দীক্ষা গ্ৰহণ কৰিও কৃষি আৰু দুগ্ধ উৎপাদনেই ব্লক বা আন যিবিলাক ক্ষেত্ৰ আছে, সেই ক্ষেত্ৰবিলাকত বৰঙনি যোগাব পাৰি বা কাম কৰিব পাৰি, সেই সম্পৰ্কে আমাৰ ছাত্ৰ ছাত্ৰীসকলৰ সৈতে এক অন্তৰংগ আলাপত জড়িত হ'ব সেপ্টেম্বৰৰ 4 তাৰিখে 7:30 বজাত হেতু দেওবাৰ সেপ্টেম্বৰৰ 4 তাৰিখ দেওবাৰে আমি আমাৰ মাজত পাম পুষ্পধৰ দাসক আশা কৰিছো আমাৰ ছাত্ৰ ছাত্ৰীসকলে অনুষ্ঠানত অংশগ্ৰহণ কৰিব আৰু তেওঁলোকে পুষ্পধৰ দাসৰ পৰা নিজ মুখে শুনিব পাৰিব বা তেওঁৰ অভিজ্ঞতাৰ কথা বা তেওঁ কৃষি খণ্ডত কৰি থকা কামবোৰৰ কথা আকৌ 15 মিনিট শিক্ষাৰ তৰফৰ পৰা ডক্টৰ বাণীকান্ত শৰ্মাক মই ধন্যবাদ জনাইছো আৰু আমি প্ৰশ্নোত্তৰ শিতানলৈ আগবাঢ়িছো ধন্যবাদ নীলকমল
Dr. Sharma, thank you very much for your wonderful talk related to vaccines, diabetics, drug development, and the role of academia in developing drugs. Amar bohuto proshno ase bibhinno platformot prothomete ami zoomot thoka proshno kitai lom so eta take proshno ami apnale agborhai jam apuni because here we can only see the questions in the Zoom chat box but YouTube or Facebook ot ki ase amar committee member e apnale agborhai jabo so prothomote Hiranyo Nathor pra eta question ase using your metaphor of the country's defense mechanism can you briefly discuss the case of autoimmune disorder in many cases doctors use steroids how do steroids help
Thank you for the question. See, the thing is, I don't have much idea about steroid medication in autoimmune diseases. But as far as my understanding, people don't even know how this autoimmune disease happens. There are a lot of autoimmune diseases. For example, as I was mentioning, that type 1 diabetes itself is considered as an autoimmune disease, but we still don't know how how these pancreatic beta cells are killed by our body itself, why they kill them. All these mechanisms are not known, but I don't know how steroid actually helps in autoimmune disease prevention. But I don't think steroid itself is a medication, but it may somehow sensitize those organs that were being attacked by our body itself to produce the proteins and whatever materials they produce for the proper functioning of our body. It may be steroids create some kind of trigger, but I don't think it's a proper medication for treating autoimmune disease.
Or just to add to that, are steroids acts as immunosuppressants and they suppress the immune system sometimes?
See, the thing is, you can suppress the immune system, but the thing is, if you already have this disease, your immune system has already worked on the body. If somebody is diagnosed with type 1 diabetes, the person is already having no beta cells. The immune system has already worked on that. So unless we know that the person is still having beta cell, immune response has not happened, then it can suppress. But once we already know that the beta cells are already killed, the body is not producing beta cells, then I don't think steroids can help in producing beta cells.
Okay. Thank you, Dr. Sharma. Next question is, how vaccines against certain bacteria work? Is the mode of action same as the vaccines against viruses?
Yeah. Both, you see, whether it is virus, fungi, bacteria, whatever is the pathogen, your body works the same way. Okay. It will produce, T cells that is particular for that bacteria or the virus. It will produce B cells that is particular for that bacteria or virus. It doesn't depend what kind of pathogen it is. As I was mentioning, our body has T cells and B cells, again, all kinds of, bacteria, virus, whatever is the name of the pathogen, it doesn't matter. We have the system in place. Only thing is that we have to activate it. So the mechanism is same.
Okay, thank you. You covered diabetics very detailed, how it works and how insulin helps and glucagon acts the opposite way, and also there was one point you have covered that there is the difference between the insulin and insulin receptor. If some people may have the problem of producing insulin from the beta cells, some people may have the problem of receptor. So receptor insulin, they have enough insulin, but receptor is dysfunctional. So if someone has both the problems, for example, someone cannot produce insulin and has the problem of the receptor also, will the injection will help, or he has to take additional medications?
Yeah, so it's a very good question. See, now we have insulin. We have a lot of insulin mimics. Insulin mimics means it is not insulin, maybe one amino acid here and there, scientists have modified. They can function like insulin. So if some insulin receptor is damaged and it can no longer recognize insulin, it may, recognize the insulin mimic. So in that case, you are not going to get an injection of insulin, but you will get the injection of insulin mimic, which can activate this receptor and open the gate for glucose. So there are a lot of, insulin mimic drugs are known in the market. So scientists have worked on that.
Yeah. So another question has come from another platform. So you mentioned that apni mention korisile je antibiotic towards your last part of your talk, because some drug companies are not interested in antibiotics, and they are interested in some non-communicable diseases like cancer or diabetics, where the drug will be there forever and people will keep on using it. But antibiotic resistance is a major problem, for example, these days. So the antibiotics which we are currently using are already, bacteria has developed resistance, and no new antibiotic have come from the pipeline for a long time. How to tackle that problem, and how some industries, for example, Novartis, Pfizer, they're still working on certain molecules, for example. But how you can get people interested, and it's still a major problem.
Yeah, antibiotic resistance is the major issue now. So most of the problem is coming from hospitals nowadays. Most of the antibiotic resistant bacteria are being discovered in hospital itself, because hospital is the breeding place for bacteria. So you may be treating yourself for cancer, but you may die from bacterial infection in hospital. So that is the major issue. See, some of the, there are different strategies for developing, molecules that are being, effective against bacteria. So other than molecules, nowadays, a lot of pharmaceutical industries are actually working on viruses and other pathogens that can kill bacteria. Okay, they're not producing molecules because molecule become resistant to bacteria become very quickly resistant to a particular molecule. Okay, but if you suppose, for example, for a case in scenario, you see, you have a bacteria that mutates very fast, but bacteria, more than bacteria, viruses mutate very faster. Virus can become resistant to bacteria faster than bacteria becomes resistant to viruses. So there are therapies now being discussed where you can use certain kind of viruses against bacteria, or certain kind of other pathogen against bacteria. See, if you remember penicillin discovery, penicillin was not used as the drug, they used some kind of fungus, and then discovered that those fungi contained penicillin. So basically, one pathogen you can use against another pathogen. So this is something being, now being developed, and now people are considering therapies in that particular direction, that you are not using a molecule, but you are going to use one pathogen against another pathogen.
Yeah. Thank you. But that virus or whatever the other pathogen should not be harmful to the human being.
Yeah.
Okay. Sayan Nandi, are you here, Sayan? Can you ask the speaker yourself by unmuting yourself? Where is Sayan?
Oh, my unmute kore disu roba.
Please unmute.
Yeah, Sayan, tumi unmute koriba parisane nije?
He said network problem. I'll read it out maybe. Okay, sir, what exactly is the efficacy rate of drugs or vaccines, as we hear about vaccines for COVID-19, that this company vaccine is 90% or this is 70%, how that is determined, and what is the efficacy rate?
See, vaccine, for example, most of the vaccines that we are using today, like the Covishield or Covaxin, it's 60 to 70%. See, this is being determined at the initial stages by number of persons you have given it, and how many percent has produced the immune response, and how many percent didn't get the disease. So when COVID was the same person who got vaccine, suppose got also infected by COVID, whether he has got the full-blown disease in respect to a person who has not given the vaccine. So when vaccine is developed, then they always work on two sets of people, and they do something called double-blind studies. Double-blind studies means the patients will not know what is being given to them, and the people who are giving the injection will also not know what is being given to them. What is not being given to them means they will give you two things. One is the vaccine, one is something called placebo. Placebo means it's kind of, without the vaccine, it's kind of a salt water. So it's kind of a negative control. So you have vaccine and you have no vaccine. And then you have, suppose, tested it in 1,000 people, 500 people you have given the vaccine, 500 people you have not given the vaccine, but only given the placebo. So now the thing is, the person who has given the injection doesn't know that what he has given, placebo or the vaccine. And the person who has got the shot also doesn't know what he has got. So both the persons now cannot, fake data, that a person who got placebo, he cannot say that he is getting a lot of infections and all, or he has fever and all. Okay, so this way, the companies make sure that there is no misreporting. Okay, and now suppose out of this 1,000 people, now if any one of them got, fever or infected by coronavirus and got the, suppose, full-blown disease, then they will monitor whether the person who has got the disease is a person who has got the vaccine or the person who has got the placebo. With respect to, suppose, a person who has got vaccine, whether the person who has got placebo is getting the disease. All these things they will monitor. And based on that, they will collect data and find out what is their success rate, whether the person getting the vaccine didn't get the disease or they got the disease, and how many. Suppose 100 people got corona after getting vaccinated. Out of those 100, how many people got the full-blown disease? Okay, because you should not be getting the full-blown disease. Suppose 30 of those patients got full-blown disease. That means your success rate is 70%. Okay, that's how you monitor. Okay. I hope I have answered that question.
Yeah, I think so. Yeah, thank you, Sayan, for asking the question. Anybody has any question from other platforms, or anybody can speak up?
Just Nilkamal.
Yeah, Pawan sir, sir, please go ahead.
Okay. Kanta, actually, I don't think whether it is a question or not. So during the lectures, if I have heard properly, so you have said that beta cell or the T cell has a mechanism to kill every pathogen in the universe. So what do you mean in the universe? Every pathogen in the universe?
This is a complex statement to make, but the thing is, anything, suppose there is a disease today in the Mars. Okay. So that disease comes tomorrow to Earth. Our body has a, cell that can tackle that. We already have that system in place. So basically, see, what happens is our body makes B cells and antibodies. So we have 20 amino acids. The 20 amino acid, if you have a 1,000, 1,000 mar oligomer of an antibody. So, at every place, you can put one particular amino acid and you can have zillions of combinations in which you can make a antibody, which will be slightly different from the other. So basically, our body can produce zillions of antibodies with different combinations of amino acids. So this is something, in chemistry, we call something like combinatorial chemistry. So our body has a mechanism of producing this, that it can produce millions of zillions of antibodies, one of which will work against this particular disease. One combination, one particular combination. Once the body knows, or once the helper T cell knows that this particular B cell has this particular antibody that can tackle this disease, then it will clone itself. That particular B cell now will multiply to billions of them. Okay, so this is basically our body produces inside our body these beta cells. Okay, these beta cells initially, even if we do not have that particular beta cell against that disease, within 1 week our body will produce it. Okay, that's why actually our body, as I was mentioning, it takes time, around 10 days to 2 weeks, to get that particular beta cell, because it has to synthesize it in the body. Once it synthesizes, it can multiply very swiftly and tackle that particular disease.
Okay, then my question was, okay, thank you for the answer, but that beta cell or the T cells, so it is with respect to all the pathogens that we know on the surface of the earth. So how do you know that there is some pathogen coming from the Mars? It is just a hypothesis. I don't think it is an experimentation.
No, experimentally we cannot verify, but the thing is, you will see that any disease you take, any disease, and you inject the pathogen in your body, you will have antibody produced against the disease within 10 days. So that's why they say that anything that comes from anything in the universe, your body has the capability to produce T cell and B cell against that. Now the question is, if the pathogen is very severe, by the time your body takes time and produce that antibody and T cell, we may not survive. That's a different issue. But if we survive the initial 10 days, our body has the capability to produce a molecule that will bind to that pathogen, whatever that molecule is.
Okay. No, my actually is whether you have any verification that there are some pathogens outside our systems, outside the earth, or outside in somewhere else. So how do you know it that there are some pathogens?
This is just a statement that I have made. We don't know that whether Mars has a pathogen. I'm just telling that anywhere in the universe, if there is a pathogen that is not there in the earth right now, even if it comes and invades our body can make a molecule that will bind there. Our body has the capacity to make a molecule against any pathogen that comes from anywhere on this universe. So because the body has a mechanism of producing molecules, millions of billions of molecules, one of them will bind it.
Okay, but I think this is just a speculation or hypothesis. This is not something that you have proved experimentally.
We cannot verify until we get that pathogen. But if you get that pathogen and inject it, you will see that your body is responding within 10 days.
Okay. Yes,
see, otherwise, you see, we get a lot of different viruses, bacteria. All the time we are being attacked by new things. But our body automatically adjusts and produces antibodies and T cells against it. Wherever this pathogen was, whether it was on Amazon or South America, wherever it is, it produces antibodies and T cells against it.
Yes.
Yeah. So as I mentioned, our body can produce, but it will take time. If we can survive the initial phase, we will survive.
But another question, this, so our beta cells, so they can produce the insulin, but even though the beta cells can produce the insulin, so we can have diabetes. And when there is insulin, enough insulin in our body, so we can have, so what's the reason?
So as I mentioned, we have enough insulin in the body, but the insulin receptor that was there in our cell, it is not recognizing the insulin. The insulin molecule, it is not recognizing because of some modification in the receptor. So now we need a molecule that closely resembles insulin, which can bind to that receptor and activate it. So insulin can be there in our body, but because the insulin binding pocket is modified, So that's why it is not now recognizing insulin.
Okay, thank you. Okay, thank you, thank you.
Yeah, thank you, Soharia sir. Next question is from Rifat Manan. Why mRNA vaccine was developed for coronavirus? Is there any type of vaccine for coronavirus? I think this was covered, but why mRNA vaccine was developed?
So basically, the idea of mRNA vaccine is that mRNA production, you see, there are a few things here. Producing coronavirus, suppose dead or alive, for suppose a billion population of India is not easy. Okay, it takes a lot of money, a lot of efforts. Okay, at the same time, for example, you take the spike protein and make a vaccine with the spike protein is also very, very expensive. It's not easy. But mRNA is something that you can develop in a lab in a large quantity. Okay, you do not even need coronavirus. You can do laboratory synthesis of coronavirus mRNA in a different, viral or bacterial cell. Okay, so it's something called heterologous synthesis of mRNA. You can synthesize mRNA. You do not have to worry about coronavirus also. You do not need coronavirus to synthesize the mRNA of coronavirus. Okay, so basically now if you are taking the mRNA of coronavirus and you produced it in a different kind of cell in a lab, and you can produce in mass quantity very cheaply. So now when you put that mRNA in a human cell, your mRNA you are putting, but you have no relation with, coronavirus itself. You are not having anything coming from coronavirus other than that mRNA. So now the mRNA in our cytoplasm can translate to the protein that coronavirus produces in its surface. So same spike protein you are synthesizing in our body itself. We are not injecting from outside. Our body itself can now synthesize that particular spike protein. So once it's fast, produces the spike protein, what our body will do is our cell will take out this spike protein and present it outside our cell, as if it is asking our antibodies and T cells to say, hey, see, I have this, spike protein. It is coming from outside. You have to destroy it. So our body will get an immune response immediately. So basically, we are not dealing with coronavirus at all, just the mRNA, but producing the immune response similar to that of coronavirus itself, just by using the mRNA. So now, because I think the major reason is producing the mRNA is very, very cheap. And second reason is it is advantageous over the DNA technology, because if you put a DNA of a foreign, pathogen, there is a chance that the DNA will integrate with our DNA in the nucleus. But because you are inserting mRNA, it is not going to the nucleus. It is staying outside in the cytoplasm. Okay, so there is no question of integration of mRNA with the human genome. So in that case, that sense also it is safe compared to DNA vaccine. So mRNA vaccine is much safer and it's cheaper. But there is an issue with mRNA vaccine is the stability. mRNAs are not as stable as DNA. So by the time you inject mRNA, if you do not have something like nowadays they are producing, nanoparticle as vehicles for mRNA insertion. If you do not cover the mRNA properly before it reaches our cell, it can get destroyed by our first line of defense itself, and it may not go inside our cells. So that is something that one has to be careful about producing mRNA vaccine, that whether the mRNA will be stable enough to reach our cell.
Okay, thank you, Dr. Sharma. Next question is from Nandi, Sayan Nandi. Do I need to be vaccinated against diseases that I don't see in humans, but can transfer from animals to humans? Like we hear about COVID-19 come from bats. What do you say about it?
See, vaccination is generally given in, this diseases which transmit very fast. Okay, something that is not transmitting, against that we don't have to get vaccinated. Okay, so only when we are mass infection starts, then only people think about vaccination, because who will produce a vaccine for something that has not infected humans? Okay, once human get infected, it gets into pandemic situation, then only people think about vaccination and all. It's a precaution, basically. But unless we are infected, nobody will spend money and produce vaccine.
Yeah. Thank you for the answer. Anybody has any other questions from other platforms? Please let us know.
আছে নেকি ৰিফাত দাদা আৰু কোৱেশ্চন?
নাই দেখা ৰে মই।
ময়ো দেখা নাই।
Yeah, so Dr. Sarma, আপোনালৈ অশেষ ধন্যবাদ থাকিল আজিৰ আমাক এই টপিকটোৰ ওপৰত এটা সুন্দৰ বক্তৃতা দিয়াৰ বাবে। আমাৰ প্ৰতিটো সেশন ৰেকৰ্ড হয় আৰু ইউটিউবত আপলোডেড হ'ব। সো কেতিয়াবা আমাৰ এইটো সময়ত কোনোবাই হয়তো এটেন্ড কৰিব নোৱাৰিব পাৰে, ষ্টুডেন্ট কিছুমানে এটেন্ড কৰিব নোৱাৰিব পাৰে, বাট উই সেন্ড টু আমি পঠাও আৰু ইউটিউবতো আপলোড কৰোঁ। গতিকে আপোনাৰ ভিডিঅ' আৰু লেকচাৰটো আৰু এই ডিছকাশ্যন সেশন বিলাক তেওঁলোকে শুনিব পাৰিব। আমি দেখিছো যে বহুত সময়ত যে ইউটিউবত আপলোড কৰা ভিডিঅ'খিনিৰ মানে viewership বেছি, because they can watch it at their time of convenience and all. So although our participants is little less today, but it was a very nice talk and we learned a lot from you regarding vaccines and drugs and how drug discovery takes place, as well as what is the role of academia and people like you who are doing research in certain areas in India and also abroad who are doing work in drug discovery and other things. So thank you very much. If Sayan and who are not the members of this, I request you to kindly leave this session. We'll have a further discussion with Dr. Sarma.